Welcome to the second seminar organized by the BRG, and it will be today regarding the biomedicine. And we will have two speakers coming from Groningen University, Paul de Vos, and then we have a case study from Shuxian. I will be the animator. My name is Denis Poncelet, and we can, in fact, start by probably give you a few information if you are not used to the Bioencapsulation Research Group. It's probably one of the biggest and most active association regarding microencapsulation. I will not go through the detail of this slide, but if you want to get more information about it, you can go to bioencapsulation.net. This web seminar is also organized in the frame of the EncapProcess, which is an European project with a large number of participants, as you can see, and it allows exchange between the participants from the different laboratory company and so on. The BRG is have this particularity that on top of organizing events, we are really supporting, in fact, such a kind of project. Then the first speaker is Paul de Vos. I know him for quite long. It's now professor at the University Medical Center of Groningen. It's a really outstanding researcher, and you will probably enjoy with me because he's also an outstanding speaker. And I will directly go to his presentation, which is recorded. Then, in fact, you can ask questions during his presentation, but at the end of the two presentations, you will have also a chance to ask questions, and he will answer directly to your presentation.
Good afternoon. My name is Paul de Vos. I'm a researcher at the University of Groningen in the Netherlands. It's in the upper part of the Netherlands. I have been in the field of microencapsulation in biomedicine for about 25 years now. And since the beginning, I'm also a member of the Bioencapsulation Research Group. I consider Denis Poncelet, who's the principal organizer of these webinars, as a personal friend. And we have been working for a very long time together. I mainly concentrate on application of cell encapsulation. The principle is quite simple. We envelope therapeutic cells, and it might be cells that we do isolate from donors, into semi-permeable membranes that do protect the cells from attacks by the immune system. And as I will explain you later on in my lecture, this is a field that is extremely popular at the moment. Also because stem cells, which can nowadays generate many different cell types, are coming onto the market for all kinds of diseases. First of all, why encapsulation of drug and cells? Why is that important? Well, it has a number of advantages over just injecting pharmaceutical agents. The first thing is that it allows for targeted drug delivery. You can bring encapsulated cells very near an organ that is damaged or diseased, and release the cargo that have therapeutic consequences quite near the disease area. But another, even more important advantage is that it allows for a minute-to-minute regulation of certain metabolites or hormones. And how important that is illustrated in the next slide, which shows the daily glucose profile of a group of patients that is suffering from diabetes. And in diabetes, the insulin-producing cells have been destroyed. And you see here that their glucose levels, which is the most important fuel of the human body, is fluctuating enormously. So what normally happens if you have a normal functioning pancreatic islet is that insulin is being released really on a minute-to-minute level, and glucose levels almost never exceed a level of about 100 milligrams per deciliter. And you see here that this is quite stable. And this is very important also for maintaining homeostasis. This kind of fluctuations, as I will explain later, can have a lot of consequences for health. Well, and that is actually where encapsulation of cells kicks in. You can transplant cells and they really sense the different metabolic agents running in blood, and can regulate this really from a minute-to-minute level so that you maintain these kind of glucose levels. The field is growing tremendously. That's for several reasons. One of them is that stem cells now can be more easily transformed into all kind of body cells. As I will show you later on, these cells usually come from certain donors that do not look similar enough to your own body cells, and we still have to protect them from rejection or from autoimmune processes. So we have to envelope them in capsules to prevent rejection. But also it has quite some industrial applications. It can be it can actually be done in used bioreactors, and that's also why many industries actually stepped in around 2010. Science grew to a certain extent that we could at a certain moment make capsules on a large scale in sufficiently high quality that industry also became interested to use these kind of systems. And I think it will the coming years still grow. And as shown here in the slide, it started quite early in the 70s, but the number of manuscripts and also the patents actually are following the same pattern as here in this slide is growing tremendously. The field of cell encapsulation is quite broad. It can be used actually in every disease where a minute-to-minute regulation of a metabolite is necessary. So that can be anemia, hemophilia, but also all kind of kidney and liver disorders. If you go in literature, you will find quite some papers about that, but also brain disorders, pituitary disorders, thyroid disorders. Even in cancer, some biological agents can really be produced quite near a tumor and in just the amounts that is needed to kill the tumors, but also, for example, in pain reduction, where you really get a release of a pain-reducing agent, really according to the needs of the patient. And yeah, if you go in literature, you will find that this list is far longer, but this is the sort of diseases in which cell encapsulation is proposed the most. Today, I will mainly concentrate on the application of cell encapsulation for the cure of diabetes. This is also the field I work in and where actually most of the scientific and industrial work currently is occurring. It's all about this clump of cells, the so-called pancreatic islets, and they are located in the pancreas. And the pancreas is just below your stomach. Every individual here online is having about 1 million of these clump of cells, and they produce insulin, but also other hormones that we need for our survival. The disease, you probably all know a diabetic patient in your direct environment, but you probably do not realize it's really a devastating disease for many individuals. Certainly type 1 diabetes, that is an autoimmune disease that usually develops at a very young age, in which an autoimmune process is killing the insulin producing cells. And these children have to take insulin injections. And this picture here is really reality for a child that is suffering from type 1 diabetes. This is the amount of insulin injections they have to take in a period of just 1 month. And this is excluding the finger pricks, which you also have to do to monitor their glucose levels. It's really a disease that is really having a huge impact on the quality of life. And you probably also can imagine that this is a huge burden on the parents of such a child. And this is why people like me are very motivated to find solutions for this disease. This slide I already showed and actually shows the issue at the moment. Patients that have to take insulin injections still have a lot of fluctuations in their glucose levels. And that can be hyperglycemia if the glucose levels are too high, but it can also be too low, which is called hypoglycemia. And if there is a hypoglycemic event, the brains will not get enough glucose, and glucose is your primary fuel for your brains. And then you might faint and you might even become comatic. If this is happening too often, your brains will become unaware of hypoglycemia. And this is actually a big threat because that can lead to all kinds of issues such as regularly fainting, but also all kinds of other symptoms may occur. And a lot of people get disabled because of this frequent hypoglycemia. So hyper and hypoglycemia is something we want to avoid. And this is where cell encapsulation kicks in. If there is too often an hyperglycemic event, the body has all kind of alternative pathways to get rid of glucose. They can become sticky to proteins, and this may actually stop up all kind of blood vessels in your extremes, like in your feet, in your hands, and on the long term also in vital organs. This is very undesired and may give all kind of issues on the long term. And this is why hyperglycemia should be avoided. But hyperglycemia is actually currently the biggest threat. So this is all the things that we know can now occur if you have too often a hypoglycemic event. And this is actually the biggest challenge that we currently have in the treatment of diabetes to prevent these kind of hyper and hypoglycemic events. And that this can be avoided just by giving the nature back to patients, like giving a transplant of the pancreatic islets, as shown in this slide. This slide shows the daily glucose profile of 3 groups of patients. Patients that are receiving conventional insulin therapy. This is how we did it till about 20 years ago. You gave a shot of insulin in the morning and a shot of insulin in the evening, but it actually never reached glucose levels that were in the normal range. And this is the normal range. This is a bit better with intensive insulin therapy. So this is what we currently do also in children, a lot of finger pricking and measuring glucose levels, and then just give a little bit of insulin. But this also never reached the normal levels like we see in patients that have received a transplant of the insulin producing cells. They are completely in the normal range, and these patients also have an almost 0 chance on developing diabetic complications, and hypoglycemic unawareness is completely gone if we can restore actually the minute-to-minute regulation of glucose levels. And this is the motivation for many people like me, and probably also quite some of my colleagues online, to find solutions to provide patients with an insulin-producing transplant. Well, with this aim, we actually have 2 options. We can either transplant the whole organ, the whole pancreas, which is quite a big gland actually, but also involves major surgery. This is actually only done currently in people that already have diabetic complications, for example, which lose a kidney because of long-term hyperglycemia. And then we also give them a transplant of the pancreas because we have to give immunosuppression to avoid rejection anyway. But it's much more elegant actually to just only give the insulin producing cells, these small clumps of cells, which are about a half millimeter in diameter. And if you all isolate them from the pancreas, which is really a big gland, then it's not more than just a finger cup full of tissue. And this is actually something we can do. But we are doing this now also since the beginning of the 70s, but we only start to understand what kind of rejection can occur since about 10 years. There's a basic difference between cell transplantation and whole organ transplantation. If you transplant a whole organ like a pancreas or a liver or a kidney, it will be connected to the bloodstream. Blood will run through the organ and sufficient nutrients will be given to the organs. With cell transplantation, that's completely different, and the same issues we meet actually with tissue engineering. We grow islets in a culture disc, or we make an artificial organ and we just give it as a cell transplant. And then the cells are going to give signals to the environment that they don't have enough nutrients, not enough oxygen, and they need blood vessels. These blood vessels come in together with cells of the immune system, and the cells of the immune system will also recognize the foreign tissue. And then all of a sudden, a very strong immune response can occur, which might result in failure of those cells. And to avoid this, we have to give very strong and complete immunosuppression. And this actually is not without consequences. If we suppress the immune system, then the immune system can also not react to, for example, fungal infections like here in the mouth, or bacterial or viral infections. And these may occur more often in patients that are receiving immunosuppression. But also, the body does not have a lot of ability to delete tumors. So if you give immunosuppression long enough, then at a certain moment, tumors may develop. and yeah, the body will not be able to delete them. So on the long term, and then you're speaking about decades, there's a 100% chance on developing tumors if you suppress the immune system in those patients. So this, of course, will never be an acceptable alternative for insulin therapy. And this is actually where immuno-isolation, so encapsulation, kicks in. So what normally happens if you give a cell transplant, then immediately when you bring it into the human body, all kinds of immune cells, antibodies actually will flow around and will kill the cells. And what we now do is, in principle, very simple. We put a little bit of an artificial membrane around those insulin-producing cells, by which immunoglobulins and immune cells cannot actually enter the capsule, while it's permeable enough to allow entry of cell nutrients, glucose, and insulin out. So in principle, it's a very simple technology. And at the moment, it's actually very fashionable, and a lot of people are working on it. And the reason for it is that we are living in the time that we spend a lot of effort on creating insulin-producing cells, but also other therapeutic cells from progenitor cells or stem cells. But what is insufficiently realized is that we can only generate these cells from certain sources. It's still not like that we can take a body cell from myself and grow this into a whole organ and give it back to myself. That is at the moment still not possible. We can only do it from some susceptible cell types, from other humans or from animals. And when you transplant them, they will still be rejected. And this is where actually we are using cell encapsulation as well. And in the past few years, I've been working a lot with replenishable cell sources from either progenitor cells or really from stem cells that started with fertilized eggs. And they really start to produce insulin, not as much as a normal pancreatic islet. It's about 2 to 10% of what a normal islet can produce, but we are slowly coming there. We are going to make, I'm very convinced, somewhere in the coming 5 to 10 years, insulin producing cells that will produce the same amount of insulin as a normal native beta cell in your pancreatic islets. And that's where we still need those capsules, not only to prevent rejection, also please remember that, for example, type 1 diabetes, but also many other diseases in which cell transplantation is being proposed, are autoimmune diseases. So they will attack the cells and kill the cells, and we have to protect the cells. And this is where encapsulation is very helpful. So this lecture is mainly about microencapsulation. There are other systems as well, when you envelope cells in larger vesicles, which is called macroencapsulation. You can connect them sometimes to blood vessels, which are called intravascular macrocapsules. But today, because it's about microencapsulation, the society is mainly about microencapsulation. I concentrate a lot on those small microcapsules, which are basically about 500 micrometers, which is half a millimeter, in which individual islets are being protected. And originally, this technology was described with a polymer called Arginate as the principal molecule to protect the islets. These polymers are actually a sort of family of polymers. One alginate cannot be really compared with the other one. It totally depends on its molecular composition. But it's a natural polymer that we isolate actually from algae. And to illustrate how different alginates can have different applications, if we isolate, for example, an alginate from an algae that is growing very slowly in very calm water, then you will see that the plant will have a very strong trunk, will grow to the surface and doesn't move a lot. From those algae, we can isolate alginate that is very high in a molecule called hyaluronic acid, and that usually gives very, very strong capsules. And we use those capsules, for example, in the peritoneal cavity or in the brain, where they have to resist a lot of shear force from organs. If the plant actually grows in water with some current, so it has to flow a bit, the plant, then you get alginates that are a little bit higher in mannuronic acid. And usually those algae are a little bit more flexible. And I use those alginates, for example, for implantations under the skin, because they have to be a bit flexible if I push my skin, for example. So this is how we can create different type of alginates. And if you go in literature, you will quite often see that people just mention, I bought an alginate, but that doesn't say me much. There are many different types of alginates which cannot be compared and have very different compositions. I will concentrate a bit on alginate to illustrate how the adventure went from very basic type of research to now something we can really apply. But there are other types of capsules as well that are being made of polyethylene glycol, for example, HEMA. There are many different types of polymers that are currently being tested and used for cell encapsulation. I'm also not claiming that alginate is the best polymer, but it's the most well-studied polymer. And we have learned a lot from how we have to go to a graft that will survive for a very long time with alginates compared to other polymers, which are less well-studied. So then come to the issues. When I started with my PhD, which was about in 1996, we had a huge issue. When we implanted encapsulated cells, they usually came out of its implantation site like this. Capsules lost the cargo, the islets, and were being covered with thick layers of inflammatory cells. And that sort of, killed the islet cells. There was not a lot of diffusion of oxygen or other nutrients, not enough supply of survival factors, and the cells died within several weeks. So we have been working a lot on trying to understand what the cause was of these kind of responses, and we have found solutions. But that was quite an adventure, and it took about 15 years before we could solve this problem. And it all started with basic studies on what is actually causing these kind of responses. And we have learned a lot. We now know that these kind of foreign body responses already start with the mandatory, although minor surgery, but it starts already there with the injury. If you make a cut, then the reaction of the body is always to release all kind of chemical signals like cytokines and histamines. So factors that will cause an inflammation. The reason for that is if there is a disruption of the barrier of your skin, for example. Also, if you do this, you will get an inflammation just below your skin, because bacteria can come in and we have to delete them. We have to keep the bodies completely safe. What then happens is that we actually get recruitment of all kind of other cells from blood vessels to delete those bacteria. And even if they're not bacteria, they still will come. And now we are doing something very strange. We do place there a strange clump of cells, the insulin producing cells, covered by a biomaterial. And whether something happens or not, whether these kind of inflammatory cells will respond, totally depends on a very small portion of the capsules. And that actually is the surface. The surface properties should be very strict. They should be smooth, not contain any pro-inflammatory molecules. They should have a certain conformation that the body doesn't recognize it. And it also should not allow too much protein absorption, because if there's too much protein absorption from just general proteins like immunoglobulins or all kind of factors that are circulating in serum, then the immune cells will find anchoring points and will release all kinds of molecules that will attract more cells. And then you get this thick layer of cells that actually will kill the islets inside those capsules, which of course is very undesirable. So we have been doing a lot of studies, and I just will give you one example to solve these kind of issues and to avoid that cells can easily adhere to capsule surfaces. So when we started with these kind of studies, there was only little attention for how the surface looked like. And it was quite often like this, that you had a sort of moon-like surface with a lot of hills and deep valleys. And on these kind of surfaces, cells like to adhere. So we started with all kind of chemical procedures, all kind of improvements of the procedures to make it more smooth. And these kind of issues you don't have with encapsulation of cells or products for food industry, but for the human body, it's very important that it's completely soft and cells cannot adhere. So we did a lot of studies, for example, with atomic force microscopy, in which you have a sort of tip of your microscope that moves on the surface and can measure these kind of changes in surface roughness. And we noticed quite soon that if we were able to make it more smooth, then all of a sudden the reactions against these capsules was close to zero. We had to do more. We also had to remove all kind of pro-inflammatory molecules on the surface and make a chemical composition, which will be explained later on, that even can suppress immunity, but we have been very successful in that. So what we nowadays do, and one of my students that will give a case study will explain it in more detail, we are now able to change surfaces as such that we bring on molecules that still are recognized by the immune system, but are in such a way recognized that they only produce tolerogenic signals. And in this way, we really can reduce responses to a great extent and almost actually to zero. So how did we do that? This is just an appetizer. One of my students will explain the whole story. But in foreign body responses, there are some receptors on cells, which we call toll-like receptors, are very important. And we have worked on different types of receptors, but we have seen that this actually can reduce responses quite tremendously. What normally happens if there is an immune stimuli, like an undesired molecule on the surface of a capsule, is that this immune stimuli will bind to toll-like receptor 2, for example. What then normally happens in the human body is that toll-like receptor 2 and 1 will fuse. This will lead downstream in a cell to all kind of responses, and finally to the release of pro-inflammatory cytokines. We have been testing a lot of different molecules, how they could interact with this toll-like receptor 2. And we found that some specific molecules like pectins, very specific type of pectins, as will be explained, can actually block the ligand binding site of toll-like receptor 2 with toll-like receptor 1. As a consequence, this fusion of toll-like receptor 2-1 cannot occur anymore. And also the downstream responses that lead to the release of pro-inflammatory molecules will not occur anymore, while the more regulatory responses that is more tolerogenic is still intact. And that actually improves biocompatibility of these kind of membranes to a great extent. This right part of the slide shows actually that the pectins are really on the surface, and that the effects are very specific for a specific type of pectins with a very low degree of methoxylation. Only then we see actually a suppression of these responses. And this will be explained in more detail by Shuxian. That will be the next lecture. She has done all those studies. I also want to take you to the inside of the capsules. This is actually something that is quite underestimated. Many people completely focus on the outside of the capsules, which is understandable because it has a huge impact on immune responses against the capsules. But we can bring that now almost to zero, and still we see that after a while, some of the islets start to fill. And we have found in the past decade that that is due to the fact that cells actually need also some survival factors when they are inside of the capsules. So normally, if cells are integrated into an organ, they have contact with connective tissue. So for example, if you take your skin, and it's a little bit flexible, that's because there are all kind of proteins, glycoproteins, that keep the cells together. And that's not only for keeping the integrity of your skin, but also they contain a lot of survival factors, and they do interact with the cells. So cells have certain receptors. I will not go into detail which they are, but they need interaction with connective tissue that contain, for example, fibronectin, laminins, but also collagen. These are called so-called extracellular matrix molecules that interact with specific receptors on those cells, and only if they have these interactions, they can survive for months. So, and if they don't have these interactions, they usually slowly die during a period of several weeks. So connective tissue is essential for cell survival. So what we also have done is not only concentrating on the outside of the capsules to make them more biocompatible so that the body will be more likely to accept them, but we also change the intracellular environment. We are still using alginates in certain compositions, but we supply also proteins that are coming from connective tissue. We use specific sequences of those proteins in order to do it reproducible. And we have seen if we incorporate them, they can have a huge impact on survival. They are a sort of spunk structure as well. They take up growth factors that they contain, which have a supportive effect on the survival of islet cells. And in this way, yeah, we have seen quite some remarkable things, and a few I want to share with you. Some of those connective tissue molecules are called collagens and laminins, and they do something strange. If cells are under attack in the human body, and that can always happen, if you have an inflammatory effect or a flu, whatever, then in your body there will be a high number of inflammatory stressors, which we call cytokines, and these cytokines can kill your cells also in the capsules. They can diffuse through. That's shown in this slide, for example. So if you expose, in this case, human cells here in capsules, these are encapsulated human islets, like shown here. Then normally, if they don't have any connective tissue proteins around, they will die for 50% in about 72 hours. But if we supply in the intracapsular environment those connective tissue proteins, and it's independent of the type that we actually give, then we see that the cells resist this inflammatory stress to a much higher extent. They will survive. So we didn't expect that on forehand. We only thought it would have a positive effect on survival and function, but also during inflammatory events that can always happen in the human body, they survive much better. So all positive things that we have learned only in the past 5 years. You cannot just only use a polymer. You also have to give some of those connective tissue proteins to promote survival. So we also did already some implantation studies, again, with some of those connective tissue proteins in capsules that reduced inflammatory stress in vitro, like I just showed, that was in a Petri dish, but also after implantation. If we implanted them, I show here a human being, but actually it was in mice, and we retrieved them after more than 2 months. We see here glucose-induced insulin release. We see that this is much better, almost 2-fold higher in islets in capsules that contains some of those connective tissue proteins. So again, a proof that we really are on the right way, and that this really has a positive effect on the survival of cells. So currently, we have implanted these new capsule types, and this is sort of principle that we apply. So we have other polymer types on the capsule surface that are sort of immune modulating. They attenuate the responses that the inflammatory cells normally have, but also we have include those survival factors, and that really has a huge impact on survival. We have still some in vivo experiments up and running, and these grafts can survive for more than a year, and we have never been that far. So it really is a huge impact on the field and on survival. I concentrated on purpose on the islet work, so the insulin-producing cells, simply because that is the largest field. But the work that I just presented has, of course, also advantages for many other applications of cell encapsulations. So actually, everywhere where a minute-to-minute regulation is required to have an optimal therapy for the disease, it will be applicable. And I just mentioned these again, where I started with in my lecture. So anemia, hemophilia, which is really at the moment a disease where cell encapsulation can be immediately applied, but also kidney disorders, liver disorders, pituitary, thyroid disorders, which is done a little bit less, but I'm convinced that also there it can have a huge impact if we apply cell encapsulation with the current generation of capsules, but also cancer and pain reduction. So I would like to end with thanking my collaborators. The work I presented was not only done by me, but also thanks to a very long time persistent collaboration with the University of Irvine of California, with Jonathan Lakey, but also with Andres Garcia, who's at Georgia Tech Institute in Atlanta, who has been of great support for this work. And of course, I also have to thank the funding agencies that made this work possible. And I'm happy to take any questions now. Thank you so much for listening to my lecture.
Okay, thanks for Paul. And the next speaker is Yuan Hu, which is working with Paul. And she's probably one of the most promising students. I saw that she already has several publications. I just put the last publication, which will be, if I understand well, and only available in 21. And she will make a study case presentation.
Hello, I'm Shuxian Hu, a PhD candidate from University Medical Center Groningen. My case study is about a novel immuno-modulatory microcapsules produced by incorporation of lemon pectin. This case study is related to the prevention of tissue responses against immuno-isolating microcapsules for type 1 diabetes treatment. The transplantation of microencapsulated islet achieves blood glucose control from a minute-to-minute level without or with less immunosuppressive treatments. However, there are still a lot of challenge limits the widely clinical application, including poor islet engraftments, lack of oxygen supply, and inflammatory responses. Here, the current study will introduce a strategy that locally regulates immune responses against microcapsules. First of all, I would like to talk where these tissue responses come from. After implantation, the encapsulated islet grafts are under huge stress. The stress comes from enzyme isolation, process of encapsulation, implantation surgery, and also the harsh environment of implantation sites, where there are low nutrient and hypoxia due to the insufficient vascularization. The stress-induced cell death could subsequently lead to release of highly pro-inflammatory molecules, also known as danger-associated molecular pattern. These stamps can be sensed by toll-like receptors, especially toll-like receptor 2, and activate a cascade of inflammatory responses, and ultimately cause pericapsular overgrowth. Severe fibrotic overgrowth will obstacle the exchange of oxygen, nutrients, insulin, and cellular waste, and finally lead to graft failure. To overcome undesired tissue responses, lemon pectin are involved in the current study. Lemon pectin is a complex carbohydrate extracted from lemon peels. Similar to arginate, both molecular backbones contain negatively charged carboxylate group, allowing cross-link with calcium ions in an egg box model configuration. But unlike arginate, the backbone of pectin consists of glycerolic acid, and most of this glycerolic acid unit contains methyl ester bond. Special procedure and conditions for pectin extraction and processing can remove this methyl ester bond, and when there are less than 50% of glycerolic acid containing methyl ester bond, we call it low-DM pectin. When the percentage is higher than 50%, we call it high-DM pectin. This chemical characteristic highly influences the biological characteristics of pectin. The ability of pectin to inhibit toll-like receptor 2 signaling has been shown in colitis models. In that study, we found that pectin, especially low-DM pectin, can bind to toll-like receptor 2 and inhibit the following cascade of immune activations. However, it has not been investigated whether pectin incorporation into a biomaterial can locally regulate host innate immunity. To answer this question, we first produce pectin arginate microcapsules by blending pectin with arginate solution together, and collected these mixtures in a calcium chloride solution. The existence of pectin on the capsule surface is confirmed by immunofluorescence staining. This result indicates that pectin is distributed through the capsules, including its surface, where immune cell capsule interaction occurs. And after that, we determine the immunoregulatory capacity of pectin arginate capsules by using a genomic modified THP1 reporter cell line. This cell line express all toll-like receptors, and I inserted construct for secreted embryonic alkaline phosphatase coupled to NF-kappa B. The activation of NF-kappa B can be assessed by measuring CR in supernatant, which is quantified by using quanti-blue. It is a medium that turns from purple to blue in the presence of CR. To test for inhibitory effect of the extracellular toll-like receptor 2, 4, and 5, the reporter cells were stimulated with agonists of toll-like receptor 2, 4, and 5 in the presence and absence of microcapsules containing pectin of DM18, 55, and 69. Capsules containing low DM pectin reduced the activation of toll-like receptor 2, but not toll-like receptor 4 and 5. The tested capsules containing the pectin of lowest DM18 showed the highest efficiency in toll-like receptor 2 inhibition. And then, human islets encapsulated in the arginate or pectin-arginate mixtures were cultured under low nutrient and hypoxic conditions to mimic harsh environment of implantation site. At 5 days of culture, the supernatant of the encapsulated human islet were collected for measuring the levels of islet-derived DAMPs, including dsDNA and uric acid. As shown in figure B and C, the amount of both dsDNA and uric acid increased significantly after 5 days of culture under low nutrient conditions. The DAMPs released was not influenced by the incorporation of pectin in the capsules. To study the suppressive effect of pectin-arginate microcapsule on DAMP-induced immune activation at day 1, 5, and 7 of islet culture, THP1 reporter cells were added to assess the activation of NF-kappa-B. As shown in figure D, capsules containing DM18 pectin decreased DAMPs-induced NF-kappa-B activation compared with capsule made of arginate alone. Microcapsules containing higher DM pectin, for example, DM55 and DM69 pectin, had less or no effect on the suppression of the DAMPs-induced immune activation. To determine our findings in vivo, we transplanted 1,000 encapsulated rat islets into peritoneal cavity of streptozotocin-induced diabetes black 6 mice. Blood glucose were measured twice a week after transplantation. In the DM18 pectin-arginate group, all animals remained normoglycemic for at least 196 days, after which the first animal became diabetic again. By the end of this study, at 7 days post-transplantation, there are still 2 of the animals are normoglycemic. But in contrast, recipients of a control arginate group and the DM69 pectin-arginate groups experience a shorter duration of blood glucose control. To evaluate graft responsiveness to blood glucose, oral glucose tolerance tests were performed. At both 4 and 8 weeks post-implantation, all mice that received islet in DM18 pectin-arginate microcapsules returned to normal glycemia within 90 minutes after the meal test. This occurs slower in arginate and DM69 pectin-arginate groups. The plasma C-peptide levels of the recipients of DM18 pectin-arginate encapsulated islet also rose faster than the rest 2 groups. At 8 weeks after implantation, we also studied impact of pectin incorporation into arginate capsules on circulating cytokines. Shown in figure A, recipients of DM18 pectin-arginate encapsulated islet had lower levels of plasma pro-inflammatory factors GL-alpha, TNF-alpha, and IL-6, and increased levels of anti-inflammatory cytokine IL-10. After graft failure, the microcapsules were also retrieved. In the DM18 pectin-arginate group, we obtained a higher retrieval rate, indicating minimal adhesion to the abdominal organs happened in this group. In the mice receiving DM18 pectin arginate microcapsules, lower than 50% of the recovered capsules had cellular overgrowth, indicating a lower levels of tissue responses. As a conclusion, this study demonstrates that microcapsules containing low DM pectin can effectively inhibit toll-like receptor 2 mediated pro-inflammatory signaling pathway, and the reduced TLR2 signaling contribute to the long functional survival of xenotransplanted islets. Thank you for listening.
Okay, I hope you enjoyed these two presentations, and it's time if you want to ask questions through the QA window, and Paul and probably Shuxian will in fact open the microphone and could answer directly to your question. Okay, Paul, you can go ahead if you want.
Well,
You can
take this question, Denis.
Yes, you can go ahead.
Okay, so there's a question about variability that you see with the pectins from lot to lot. So in fact, what we do, just like with alginates, we everything we get in is being tested for composition. So especially with the low DM pectins, we know there's a certain range in which they actually suppress toll-like receptor 2, so the central immune receptor in this case. And we select this type of pectin then for these kind of studies. And also we purify the pectins ourselves. Shuxian does that in order to remove, for example, endotoxins. So we know exactly what kind of composition it has. And if you go into our papers, we have one paper of Shuxian, but we also have a couple of other ones where you can see how and to what extent the pectins are being characterized.
Okay, any other question?
Okay. Yeah, there's another question about diffusion of the nutrients. So these capsules are somewhat porous. What is the porosity? Yeah, there's still a lot of debate on what kind of permeability you should end up with these kind of capsules in order to provide immunoprotection. It is a bit of a balance. So you want to have some larger molecules still to flow in for survival. So for example, transferrin, which is an iron carrier, which is quite large, is about 80 kD. You want to have that in. So we usually aim to have a sort of permeability in which molecules up to 100 kD, so a kilodalton, can still diffuse in. But it's not that simple. It also depends a bit on the conformation of the molecule. So we also look into that. But we try to allow that, and then everything above 120 kD should be limited in diffusion, because most immunoglobulins like IgG, they have a molecular weight of about 160 kD, and you want to keep them out. So usually we try it's a process in which it's not extremely strict, so it's always between 100 and 120 kD. And also dependent on the concentration of the molecules outside of the capsules.
Okay, any other question? Okay, in the Q&A, we have two questions. Paul, maybe you can answer quickly, and then...
This, in fact, were the questions from the chat. Yeah. So what is Q? Oh, sorry. I see here Q&A. I have to open this. Did you measure HP-sec mole, the molecular weight distribution of filtration in UV? So Shuxian's answering, we do that for alginate, has not been done. Yes, has been done for pectins as well. Yeah, could you comment on the stability of alginate capsules after in vivo implantation? Yeah, so up to, we have done these studies. So in the studies of Shuxian, we retrieved them for periods up to a year, and then they are still very stable. Something we didn't discuss in a lot of detail, but both the alginates and the pectins, they can bind calcium according to the egg box model. So hyaluronic acids, parts of the polymer are being bound, and that is quite stable. So there is a sort of equilibrium at a certain moment, but yeah, in small rodents, we went up to 2 years and still found them back completely intact. We have also done in the past, a couple of years ago, some studies in pigs, and we could retrieve them after up to 3 years without changing their conformation, kept about the same capsule diameter. So they're pretty stable. I know there are some papers about instability, but that actually is quite dependent on the system that you're using, the concentration of alginates, what kind of divalent cations you're using, whether you use polyamino acids to stabilize. And yeah, so if you do it in the correct way, they can stay stable for many, many years. And there are even reports of trials in humans that they were able to retrieve them 8 years after implantation in a human being. Another question is how and where did you implant the alginate microcapsules in diabetic mice? So in these studies that were presented by Shuxian, it was in mice. You can take it as well if you like.
Okay, yeah. So for this study, we implanted all the microcapsules in animals' abdominal cavity, and we first make a cut near the linea alba and use a syringe to inject all the capsules.
Okay.
Yes. Yeah.
We also have done some studies, but not presented today, subcutaneously. They stay stable there as well. We have experienced that we can induce normoglycemia, but because the vascularization degree on the skin is lower than in the peritoneal cavity, the capsules and cells tend to survive for a shorter period of time. So that's why we still favor the peritoneal cavity. But we have also projects in which we try to improve the vascularization degree on the skin and therewith also supply of blood and nutrients. And yeah, we hope in the future we can also use the skin as an implantation site for these kind of microcapsules. Another question is, could this type of treatment be affected by variability amongst diabetic patients? Yes, of course. It depends, of course, to the degree of how diabetic people are. If they are diabetic for 40 years and have a lot of metabolic demands, it will put in far more higher pressure on such a graft. And I expect that these kind of grafts will survive for a shorter period of time. But in general, I still feel that we can do this actually in a very large group of diabetic patients. Then there is another question, encapsulation efficiency and stability. Well, that has been sorted out over the past 2 decades. The efficiency is almost 100%. We don't lose a lot of cells. You must imagine that the cells that we are using are donor cells coming from cadaveric donors, so people that passed away. So there are also ethical issues there that we have to be very precise in what we do. So we really treat that tissue in a very efficient way, and we don't lose anything anymore. So production rates, for example, in our case with microcapsules is compared to industrial applications quite slow, but we have to do that in order to treat the tissue well. Now a lot of questions come in.
We'll answer 2 or 3 more questions, and then the people will contact you for the future question.
Okay. Could you consider that it could have some future for capsules with vaccines? What could be suitable capsule size for vaccination? Yes, absolutely. For controlled release of slow release of some vaccines, it might work. Yeah, absolutely. Yeah.
have you defined a minimum thickness necessary between the capsule islet and the outside of the capsule that's necessary for immunoprotection? Yeah, that's a very good question and very difficult to answer. In our case, we really have seen that if cells are really in the edge of the capsules, that they can still provoke responses. And it is for two reasons. The first is that they sometimes even can protrude. So some capsule components really can stick through. But what can also happen is that if the capsule is too close to the surface, then the surface characteristics might change. So for example, you have more roughness, which inflammatory cells can adhere. We always try to keep them around 50 micrometers inside of the capsules, but it's a phenomenon that we have discussed a lot. And honestly, we do not know the answer, whether they really need that 50 micrometers. We never see actually a lot of responses if we keep that strictly, but it might also be 20 micrometers. Simply do not know. So another question is from Dr. Sopena. How long are the cells viable in microcapsules? How measure viability of the cells in the prospective treatment of diabetes with the therapy? Would you need reapplication of these? Yeah, okay. So that's also a very good question. I have had a lot of debates with insurance companies, and insurance companies will apply these kind of therapies if we are able to keep these kind of grafts functional for about a year. Only then it's profitable for them to finance these kind of therapies and to provide it as an alternative for insulin therapy. And we can do that now. We now are at the stage, at least in animal models, where we can keep them viable and functional for at least a year. So that makes it then all of a sudden attractive also to apply this in the future for human beings. So that also answers your first question, how long can they stay viable? Well, in our hands for at least a year, and Shuxian has still a number of animals that are normoglycemic, so they can run longer. And we have different measures to measure viability of those cells. We can, of course, quantify after retrieval, but we also have islet cells that are bioluminescent. That means that as long as they are viable, they actually light up. So we have studies still ongoing where we have islet cells that light up in animals, and you see that there is a gradual decrease in viability after a while, but even after a year, we see many viable cells still in the peritoneal cavity. So my answer should be now at least a year, but yeah, it is probably longer. And then on the other chat, it now all of a sudden becomes quite busy. They lose viability over time. Is there a risk of sensitizing the patient to some graft antigens and reduced viability for the second and third transplant? Well, that's the principle of immunoprotection. They should protect the graft. So even if you sensitize, which happens to a certain extent, we have looked into that might happen indeed, then the capsules should still protect the cells for these kind of sensitization that happens. And we have done in the past repeated transplants, and they can actually survive again and again for the same periods that we initially had. So it doesn't seem to affect the islet cells too much.
Okay, I think we will let people ask the question. You have the email of Paul and Xiang, and you can we may go to the end of this web seminar. You see here that we have planned it, but we will see if we can keep it at this date, an industrial convention and a conference for next year. And we'll have also a new web seminar. You will find the information on the bioencapsulation.net. If you go back to the web seminar for the biomedicine, probably over, let's say, one day, you will be able to recollect, in fact, the podcast of this web seminar. I would like to thank all of the participants, especially Paul and Xian, for their presentation and their contribution. And I hope you will have a good end of year, a happy Christmas, and that the next year will be a little bit easier than this year. Have a good day and see you soon.